From Microbe TV, this is This Week in Neuroscience, episode 52, recorded on June 10th, 2024. I'm Jason Shepard from the University of Utah, standing in for Vincent, and you are listening to the podcast about the nervous system. Joining us today is Tim Chiang from NYU.
Hello,
How are you doing, Tim?
Welcome back once again.
Also joining us is Vivian from in New Orleans. How are you doing, Vivian?
Doing great, thanks.
And then finally, we have special guest Mauro Costa-Mattioli. He's currently at Alto's lab in San Francisco.
Hi, guys. Always a pleasure to be with you guys.
All right. So today, Mauro is going to talk about his latest work on autism and this paper that was recently published in Cell, Host, and Microbe. The title is Precision Microbial Intervention Improves Social Behavior but Not Autism Severity, a pilot double-blind randomized placebo-controlled trial. So this is an actual translational work that you're involved in now, Mauro. So not just mice.
Not just mice. You are absolutely right. Probably some of you might remember, but in 2016, we have that discovery that a single microbe is able to reverse social deficits in animals, in animal models. At the time, it was quite interesting discovery. Some of my colleagues, I think the microbiologists, they love it. The neuroscientists, not quite so. How come a microbe could affect the brain, and so on. But we stuck to it, and we continue trying to figure out how this microbe is working, whether this was applied to different models, and the answer is yes to all of this. But we never expected that someone will go and do a trial with this microbe, right? And a colleague, Luigi Massoni in Italy, he ran this trial during the pandemic, actually, and he called me. I vividly remember the call. He said, "Mauro, there's something wrong." I said, "What is wrong?" I said, "The microbe. The only thing that fix is the social deficit." I said, "What? That's the only thing that's supposed to do, right?" So anyhow, so it's a long story short. I actually called some other colleagues from Stanford. I wanted to make sure that the findings were actually accurate. We did some experiments with other strains, but it's still quite remarkable, right? That after just a few years, you can move to the clinic. And as we describe on the paper, while our paper has been under review for more than a year, to be honest with you, there was another trial that was done, and the outcome is actually the same. So there are two trials now. So so I mean, I think it's an incredible time. There's so much to do now in the future, but we are incredibly excited that those findings in mice, and everyone tells Jason, mice are horrible to translate. Maybe if you have something good, they're not so bad, right? So we are very excited about be able to change behavior in kids that they have this disorder.
But yeah, just to set some background to what Mauro is saying, Mauro, you're actually last on. I'm checking through the old episodes of TWiN podcast. You are last. You're returning, our first returning guest, I think, and you're last on 3 years ago to talk about your cell paper exactly on this microbe that showed. Just to give some background to the audience, where you found that giving this microbe to a mouse model that has autism, it actually rescued specifically the social deficits of these mice, but not some other autism deficits such as hyperactivity. And you said it's taking you a while, but I have to say this is the first time on the show where someone has shown kind of bench to bedside application of the finding, and to do it in 3 years, I think it's remarkable.
It's amazing.
So
Actually.
it is. I mean, it is amazing that of course, as we go by, I mean, I see microbes like a bag full of chemicals, and if a chemical is able to change the brain, my bag full of chemicals should be able to change the brain, right? So, but it's still, it's sort of, 10, 15 years ago was sort of inconceivable, that idea, right? That you can have a microbe that can be used to essentially change, the behavioral state of a human, right? I believe that this is the first demonstration that you're changing one of the core symptoms of the disorder, right? Which is the social deficits. With microbes, you can change, the flora of the gut, and if you have, other symptoms which are not core, maybe aggressiveness and so on. But a core symptom, I think this is the first demonstration, and the fact that another lab does, another parallel clinical trial, and the outcome is actually the same, I think it speaks very highly of the potential that this might have in hopefully in the future, in the near future, I would say.
Yeah, I was just going to ask if one of the questions I had at the end of reading the paper was if you and your colleagues and maybe a growing population think that kind of gut microbiome profiling is going to become part of the diagnostic and treatment process for individuals with ASD.
Yeah. So there's several important messages. So there's the translational aspect that comes with 2 strains. So it's a pill that contains 2 strains of Reuteri, of that bacteria. When we go and we split those strains in individual strains, we found that only one of the strains is the one that is driving likely the effect in humans. That is, strain A has an effect in mice, strain B does not have an effect in mice. Presumably, we are diluting the effect. So as we think about advancing this as a potential treatment, I think it would be the next trial. It should be focused on the strain that has the effect. Regarding your question, Vivian, is we were thinking in taking responders versus non-responders and see whether the responders, they have a particular microbial profile to start with, which could educate us in a future trial just to take those guys because that they will respond. So, but we don't know whether that's the case, but I think it would be very interesting, just building on what you're just saying, this is sort of a reverse trial, right? You do the trial and then you analyze the data from the trial that will educate you for the next trial. So this is something that we are considering, and hopefully we can move forward doing it this way.
So when we go through the paper itself, or go through the trial and walk us through what they actually did. I mean, I think this is interesting. We generally have covered, classic basic science papers. So I think this is one of the first sort of clinical trial papers. And I think that could be interesting for the audience to sort of see how they work.
Right. So, and of course, I will make a lot of mistakes because I didn't do the trial, right? So, the trial was done by my colleague in Italy, and I will tell you what I know. But if the audience have questions, please send it to me, and I will send it to Luigi in Italy, and he will likely reply. So, there's a recruitment of, in this case, kids with autism. I think it's between 4 and 8 years old.
I think it was 2 and 8 actually.
Oh, sorry, 2 and 8, 2 and 8.
But the
Yes.
mean, but that mean, it was like between 5 and 6 years old was what most, was the
Yeah.
average.
So, so then what it actually is done is, okay, so the trial was designed by Luigi, and he put as a primary outcome autism severity, okay, which in retrospect, the primary outcome should have been social behavior or social deficits, okay?
Right. And actually, this is a stickler. So I remember when the Fragile X
Yeah,
syndrome trials were going on,
yeah,
and I was involved with peripherally because of my postdoc advisor, Mark Baer. But
yeah,
part of the issues when a clinical trial fails is because the outcomes weren't accurately, I guess, um, identified in the first place.
right, right,
Right,
right,
operationalized in the wrong way.
right. Well,
So like oftentimes parents will say, "Oh no, it worked. The drug worked for my child," and of course you have to get rid of the bias and placebo effects. But sometimes a lot of the symptoms that were maybe actually affected weren't in the actual outcomes.
right. So there are measures which are taken. So now you have a group, let's say 40 kids. There is a basic measure that is made before treatment, and then they will get randomized. This is double blind, so the experimenter doesn't know the treatment, uh, and the patients or the individuals, they don't know what they are getting. Okay, and so they are given the bacteria or a pill which doesn't contain nothing, essentially twice a day, and so there are measures which are being made, and the last measure is made at 6 months. Okay, so in order to measure the so-called autism severity, there is a test which is called ADOS-2. That according to the clinicians that I spoke with, in order to move this, in order to have a positive outcome, you need a bulldozer. You need something that is going to be like a bomb, right? And this is a test which I've been told it is not very good at measuring changes in the short term. So if you measure, let's say, autism severity over years, it's actually much better. And then there is essentially the social test, which there's different measures that you can take. And actually, Antonio Harden at, uh, at Stanford, which is also a co-author of the paper, essentially he has developed different ways to analyze social behavior in a more specific way, right? It's like, this is not, when we think about individuals, this is not like animals that you just put them to socialize, right? When we do experiments with mice, so, social behavior has so many other components. And when these different measures of social behaviors are made, uh, uh, then the data are put all together and they are analyzed. And this is where, in those two measures, we found that the bacteria was effective. The bacteria was not effective to change the ADOS, the so-called autism severity. I would have predicted that would be the case. But again, the trial was designed this way. I think if you were to have as a primary outcome social behavior, the paper would be probably in New England or in Lancet.
Right.
But that's okay. I mean, the good thing is that, as I mentioned before, there's another trial that shows the same. And as we think about the future, the primary outcome should be social deficits, one
Right.
of the symptoms of the kids with autism. Um, so this is essentially, the beauty also, which is double blind. There's no bias. I have to say, though, that the social tests are sort of observational and questionnaires that the parents feel,
Yeah.
right? So
Mm-hmm.
so now when we think about another trial, I would have preferred, and again, this is we didn't raise money to do any of this, Jason. I mean, we didn't have a grant and anything, right? So
Yeah.
as we think about the future, uh, I would be more confident if we have one person which is analyzing the kids come to the clinic and is actually analyzing the behavior before and after.
Yeah.
Okay.
I mean, I've heard this before as well. Same with the Fragile X, where it's if parents are filling in the survey, they want this to work, and that's
Yeah.
often the case that they're sort of biased. They'll sort of, it could certainly creep in.
Right.
But it's double blinded, so hopefully that gets rid
And
of that.
placebo.
Well, yeah, it's double blinded, but again, I think when we think about the future and we want to think, this is if this were to have an effect in ultimately in kids. I mean, I think it is going to be quite relevant, right? So you want to have a right. So
Yeah.
as we
No, I
think
mean,
about the future, I think we should include this additional it's a test of rigor, right? It's a different test of rigor that will reassure us that uh,
yeah.
that actually we are having an effect. And now an independent observer, a clinician which obviously knows about behavior in social behavior, can look at it in an objective manner and tell us. I think that this will be, as we think, how can we make it better? This will be something that I think it should be done.
Right. Yeah. I mean, so like the double blind part. So I'm a little confused by that. So the parents fill out the form, the behavior survey, but that's
Mhm.
not blind. They're not doing it for someone else's kid, right?
No. They do it for their own kid, but they don't know
Yeah,
whether they receive placebo or they receive.
exactly. So the problem is that, and I've seen this directly, is that there's so much hope from the parents for something to work that
Yeah,
clouds their observations,
totally,
or it
totally.
can.
Right. No. It can if you think about it objectively, right? So let's say, so I tell my people, they don't like me to give that example, but I tell my people, okay, I want to know an answer today, right? So let's say we die tomorrow. I want to know, tell me what you think you can see in your experiment. So let's say we die today. I will say, okay, very likely today I can tell you it likely work. Why? Because the parents that they gave placebo, they also want to work, but didn't. Okay.
Right.
And there's a second trial also that shows actually an improvement. So today I would say when we build on the next trial, I think we feel confident that this uh, could be reproduced, and if we find and we get the best, the strain that works, maybe can be enhanced. Now, something that we haven't been able to do that this is that I hopefully will be the last chapter is what I haven't been able to do, Jason, is I haven't been able to identify what is the social gene, what is the gene in the bacteria which is driving this.
Mm-hmm.
Okay,
Mm-hmm.
so we are very much interested in trying to figure that out because there is a potential that if you were to have that, now you put it in 3 copies or in 5 copies or in 7 copies, and can you make a super social bag, a bag that will be way more powerful, right? So now, if this were to be the case, now a new trial has to be done
Right.
with this bag. Yeah.
Do you
It's
think?
interesting. I was just going to say, it's interesting that you say that can we make a bag of chemicals that's even more potent? And so it's really going to, you could even like titrate it based on an individual's severity. But I noticed that in one of the figures that I don't know how we're going to go through the paper, if we'll break it down, figure by figure and go kind of chronologically. But when you have your mouse model of autism that you use in this paper, and then you give it vehicle or you give it that cocktail of Ruteri, you actually the values for time that the mice spent interacting with the conspecific or the novel the conspecific versus an empty cup in the 3 chamber, which
Mhm.
we've talked about before on the shows several times. So I hope that listeners are familiar with that. But they almost appear hypersocial in a way. Like
Yeah.
that analysis, those statistical comparisons were not, if they were done, they weren't presented here. So we can't really say what that means. But I'm just looking at the raw values and thinking, wow, that's really interesting that we may
Yeah.
have we're actually like driving a desire. But this is in contrast a little bit with the findings, like you were saying, that social skills can be broken down into multiple domains. Social communication, I believe, was the one where we saw the most improvement in the human patients, well,
Mhm.
the subjects. But social motivation and was not significantly altered. So I
Mhm.
just think it's interesting to think about like this increased sociability. Potentially, I know that has not been statistically shown here, but the increased sociability that you see in the mouse model that received the Ruteri is increased, and I wonder if is that social motivation in the mice being affected? Is that what that?
Yeah. So I don't know, but my understanding, and I think the best person to answer that question would be Jacqueline Crowley, who developed that task, the free chamber task. My understanding is, so for the audience, so essentially uh, you have you know animals in which there's essentially three compartments. In one on the left, there is sort of like a cage in which there's nothing, and on the right, there is a mouse. And now the animals which are social, they will spend most of the time on the right, in other words, interacting with the mouse. Whereas the asocial mice, they will spend equal time between asocial and social compartments. Now, my understanding that this is a task that would allow you to tell whether you're social or not, but whether it cannot tell you whether you are more or less social. In other words, it's a yes or no task.
Hmm.
So if it's statistically significant, if you have a difference which is statistically significant between the groups, and the difference is big or small, you are social.
Mhm.
That's my understanding. My understanding is that I don't know why it's so, but maybe you can ask Shaki to be in one of your episodes and explain you this. So just to start, so we never pay too much attention when we see. Uh, well, of course, we like when we see big differences, but this is what biology is, right? So now, so in that task, in the mouse, whether this is social motivation or social reward, or this is more salient for the animal, we don't know. Something that we publish, and actually we have shown in every single of the models of social behavior that we have found, is that when we put two animals to socialize, and we uh, chop the brain after, and we record from an area that sends reward, we find that those neurons, which are dopaminergic neurons, are they actually, the connection between those neurons is actually strengthened.
Hmm.
Animals that are social, you put them to socialize and you do the recording and there's nothing.
Hmm.
Let me go back. This increase in social reward is similar in firing of those neurons, is similar to the one that an animal will have if you give them cocaine, amphetamine, or any other drug of abuse, as a matter of fact. So we believe that um, in part, maybe the lack of social behavior might be due to the fact that for those animals, social behavior is not a rewarding experience. Why should I interact in the morning
Yeah.
with someone if I don't have a pleasure? I don't have any pleasure to interact with someone, right? So, can we deconvolve this in a trial in humans? We haven't discussed it yet, but whether we can devise a task in which assess social reward in a better way.
Hmm.
And
I don't think that no one has done it, to be honest with you.
yeah.
Hmm.
I mean, are there other, so in the mice at least, did you look at other kinds of rewarding experiences? Is it really specific to social?
Yeah. So if we go, so those animals that you put them to, okay, so you put the two wild type animals to socialize, boom, you take them, chop the brain, do a recording, you see activity. Okay. Um, in all models, genetic models, Shankfree-B, CANAP2, maternal, in all those models which have social deficiency, that they have an impairment in social behavior, you put them to socialize, and there's no change in the firing of those neurons. You take those animals, but now you give cocaine, boom.
Right.
You give amphetamine, boom. Right. So
Right.
it's specific to the social circuitry, and the way what we found, and just to tie it with all the other papers, what we found is that uh, uh, so our data supports a model in which what the bacteria does is trigger oxytocin production in a particular brain area, which is called the hypothalamus,
Mm-hmm.
in a particular brain area of the hypothalamus, which is called the paraventricular nuclei of the hypothalamus.
Yeah.
And actually, there are those oxytocinergic neurons. They do a connection with dopaminergic neurons in the reward center. Okay, and after our original paper came out, Rob Malenka from Stanford essentially has specifically activated or repressed the activity of those neurons. And he has found that he can promote or inhibit social behavior specifically.
Right.
Mm-hmm.
So that's, I'm sure you were thinking how this can possibly be specific. I think that's where the specificity lies. Is
Right.
this that circuitry which might be deficient?
So it's through the oxytocin pathway. So for as a reminder, oxytocin is a neuropeptide that has been already implicated in social behavior. I mean, it's one of the poster childs for sort of modulating social behavior, at least in the context of this monogamous versus non-monogamous pairing. This came out of prairie voles. This idea that so some mice, they'll be they won't mate for life. They'll was not mice but rodents.
Voles.
uh,
Voles.
and even some voles, but they're the prairie voles, I guess. They mate for life, and the question was what was going on there, and it's through the oxytocin pathway.
Yeah, and this is, I think this is probably is a central pathway for social behavior, right? So there is kids with have loss of function mutations in oxytocin receptors, kids with autism, so there's a genetic component. If you were to use oxytocin, if you were to puff oxytocin, so the question I'm sure you guys are thinking is why we need a bacteria, why we don't use oxytocin. Okay?
Yeah.
So if you just give oxytocin, and the way that oxytocin is given is intranasal, and the amount of oxytocin that you give is massive. Okay, so we have done it in our experiments head to head with the bacteria, and you give oxytocin, it actually works, but the effect essentially wears off very, very quickly. It's like you give oxytocin an hour, immediately you test it, the animals are good. An hour later, 2 hours later, the animals, they are socially impaired again. Okay. So what happened is that as a result, if you were to give it to infants or kids, the amount of oxytocin that you give is massive. You need to give it essentially almost every hour or so. Receptors get internalized, given such a massive amount.
Does oxytocin cross the blood-brain barrier?
Well, it does in the animals.
Okay.
It does in I think in patients also it does,
Okay.
but not very efficiently. So that's why you need to give, or at least when you use the intranasal route. So as a result, it doesn't work. Actually, there's clinical trials that they show that actually oxytocin ultimately doesn't work. The reason I think why it doesn't work is because what we believe is that with the bacteria, what we are doing is you're increasing the activity, the endogenous amount of oxytocin, sort of in a persistent way,
Right.
rather than have that peak and then, So now, if we were to have an oxytocin that have an increased half-life, that would answer the question.
Right.
Right.
And so you still think the bacteria are producing some metabolite itself that somehow activates this endogenous oxytocin pathway.
So in the previous paper that we discussed here, I don't believe we discussed the original paper, the 2016. We discussed a 2021 paper.
Right.
Both of them were in Cell, but I think that's what we discussed. When we did metabolomics in those animals, what we discovered is that normal animals that they have social defects, when we look at the metabolomic profile from the animals that have a social defects because they have a genetic mutation, is very different. Okay. When we put the bacteria, we didn't reverse the entire metabolomic profile, the metabolites that the animals have in the gut. But when we look at the specific metabolites, we found the metabolites from the biopterin pathway. Those, they were reduced in the animals and they were reversed by the bacteria.
Uh-huh.
And now, we can block the synthesis of biopterin, and we can show that the bacteria has no effect. And we can also do the opposite. We can just give biopterin in a bacteria-independent fashion. And we also reverse the social deficit. But like oxytocin, biopterin is oxidated very, very quickly. Biopterin, which is exogenous versus endogenous, the oxidation is like 5 times faster. Mm-hmm. So you are also dealing with an issue of, but yes, Jason. Now, is biopterin produced by or induced by the bacteria? We don't know yet. Uh-huh. Okay. But yes, we believe that there is something in the bag, because there is something in the bag, which in the bag that has an effect, which hopefully in the genome, obviously, there is something that is allowing the bacteria to have this incredible effect.
And you still think in autistic kids that they're lacking this pathway? I mean, I think this is what's remarkable to me is that, these patients were not, they're pretty they're from all walks of life. They're not a monogenic disorder. And so, why is this a common deficit?
The answer was a common deficit. Is because it's affecting probably the central pathway that regulates social behavior, which to me is from all the
But
evidence.
I mean, in terms of, is it that all these patients don't have this gut pathway, or is it just that the drug affects that central circuit?
Yeah. So just to be clear, the only way that we can demonstrate that the absence of the bacteria, and I will go back to this. The bacteria is very difficult this strain, and even is very difficult, Rutra is very difficult to find in the population. Uh-huh. Has been eliminated from the population. Huh. Okay. If you go to natives or, isolated populations, you might find it. Okay. So, when the first paper came out, there was a big argument that people say, oh, it's because you don't have the bacteria, you have social deficits. What I am confident is the opposite. I'm more confident that we can use this gut-brain axis or microbial gut-brain axis as a way to affect the brain.
Right.
In an effective way in animals and in humans. Mm-hmm. To demonstrate that it's the absence of the bacteria which drive this, Jason, you need to go to a mouse. Let's go to a mouse first. You need to go to a mouse which has the bacteria and get rid of it, only of that bacteria, and essentially show that the animal has social deficits.
Yeah.
And I don't think today we have the technology yet to do it. We can do CRISPR and kill a bug, but in vivo, you can reduce it, let's say 2-3-fold, but completely eliminated in vivo, it has been difficult.
Right.
So there's a lot of people which are working towards this. There's incredibly interesting advances on how to do that.
Mm.
So, um,
we
so
do know.
go
Sorry,
ahead.
but we do know that you found that in germ-free mice, they do have social deficits. So it suggests that, a collection of bugs is important for social interaction. And I guess the question that you're trying to ask is this single bug
Yeah,
important out of all of them?
yeah, but but this, what Jason was referring to is that you have all those different etiologies, right?
Mm.
Bugs and no bugs. It could start from the microbe, could be a developmental process, but if this were to be a process which is exquisitely sensitive to an insult, right? So now the system shut off, and as a result of it, uh, maybe this is why you know the the animals, at least, they have these social deficits. We remove Shan3B, we remove CANAP2, you put high-fat diet, you have models of idiopathic models that you don't know the reason why the animal has this. Invariably, in all those models, when they have social deficits, when we look at the recording of those neurons, we find those deficits as well, right? Um, so, and if this were to be the core of social behavior, right, at the molecular level, Jason, if you were to be if you are able to modulate it, I think this could explain why it is effective. Now, if you have a developmental problem and the circuitry is broken, I would expect that the bacteria will not work there because so that will essentially explain why it might not work in all cases.
Yeah.
And B, you may have other dysfunctions, right? There's also other molecular processes that get miswired and that also lead to social deficiencies. So, but at least if you can tackle a population, right? I mean, I think that would be awesome. When we start to characterize all those animals of social deficits, our goal was to see whether we can do precision. We can say, okay, let's say patients with Shank3B, they don't respond. Okay, out of the trial. Okay,
Right.
or others they do respond. Okay, boom, let's use those, right? So that was our original intention. But the fact that it's safe also, I think this explains also why I accelerate things
Right.
tremendously.
Yeah. I mean, I think in the end, that for therapeutic purposes, it doesn't matter if it's because it's replacing a bug or it's just hijacking the circuit. But I think, as you said, like if you could narrow down the population that it works the best in, then that obviously would also help.
The other aspect that is important to mention is that the bug has to be supplied constantly, every day. Okay, so our experiments in animals, we use a human strain, right? The same that very similar to the one that we use in the trial. Um, and, um, and the reason is that the bug will have a hard time to colonize. A human strain will have a hard time to colonize mice. But I think even a human strain will have a hard time to colonize when you have ecology which is already established. So let's say I want to change myself. I want to change entirely how grad school functions in Utah. Okay, so there's two ways that you can do it, right? One is you come and let's say you are Mario, or you you win the Nobel Prize, and you come and people will have to listen to you. So you just throw a bomb. A bomb comes, which could be, in this particular case, could be antibiotics, right? So now you just put a bomb, and now a new community is generated. Or the other way to do it is through Jason. If I talk to Jason every single day and I give him my ideas, and those ideas they get to whoever in Utah, if you are persistent, that's the other way that you can do it.
Right.
So the way that I envision, unless you also can engineer the bacteria to help the bacteria to colonize. Okay. But as such, my prediction will be that we'll have a hard time to colonize, the ecology, an ecosystem which is already stable in the kids, in humans. So that's why you will have to give it every day.
So
Right.
since it's hard to get the bacteria to colonize the patient, do you have any data, or does the Italian group has any data after the trial? Do they get back to the kids who got the bacteria get back to the placebo group's level, or is it a
So
persistent?
they look immediately after, and of course, actually, there's a figure. I don't remember. You may have the figures. There's a supplemental figure in which you can essentially clearly see that the placebo group there is zero bacteria, 0, 0, 0, and in all the kids you have different levels of the bacteria. I know that you will ask this, Tim. You say, "Okay, Mauro, can you correlate how much bacteria a kid had?" And we look into that too, but we don't have the power. We don't have the power with 20 kids or so. We don't have the power to do that analysis. The other question could be: Can you look at kids which are high functioning versus low functioning? Do the high functioning respond better than the low functioning? If so, the next trial should be just with high functioning, right? So we have some idea, but we don't want to conclude because it's risky when you have such a small number. I mean, it's a good number to start with, right? One of the reviewers asked, he wanted more patients, right? I said, listen, this is not like, Jason, when you revise your paper that you can do more experiments. I cannot go back and put more.
Yeah, for mice, you can like add in, but for
exactly
humans,
right. So it is right, and then of course, it's a first trial. People wanted to have 200 kids. Say, well, I mean, who is going to support this? Who is going to fund this, right?
Yeah.
So now you gain confidence. We have some ideas how to go about it now. We are discussing with different people, and
that's what I was going to ask you now. So I mean,
I know there's some companies probably that would be happy to support
we
it.
didn't mention this, but Mauro used to work for he was at UCSF. Before that, he was Baylor College of Medicine, but now he's at something called Altos Labs. So maybe you want to tell us a little bit about that, Mauro.
Well, so this is from the financial standpoint, it's going to be very difficult because so those strains are, okay. So we screened. Let me spell this out, and this will be more. We will need someone who really wants to have an impact in society rather than making money.
Yeah.
Let me explain you why I think so. So the Reuteri, you go and you Google Reuteri and you have companies that they sell this, they sell that, whatever, you can find it, We took many different Reuteri strains, okay? Strain 1, 2, 3, 4, 5, whatever, right? In the paper, we have two human strains, one that works and the one that doesn't work. But we took a bunch of them. We took strains from birds, Reuteri strain from birds, Reuteri strain from pigs, also from rodents and so on. And you know what, Jason? Some of the strains, which when I talk to bacteriologists, they say, of course, but for me, it was very counterintuitive. Some of the strains work and the others, they don't. Okay? So now you have your bag here, there. So now let's say I want to get an investor who wants to support this. They will say, are you kidding me? I go to the supermarket and I get a strain. They don't know whether it's going to work or not, right?
Yeah.
So unless you find the gene, or unless you have someone which essentially can have an impact in society, and I am all for it, to be honest with you. I think this is, there's so much promise, and there is this new movement in neuroscience which is gaining this momentum that you can essentially affect the brain by structures which are far removed from the brain, or even by cells, Jason, which are not neurons, right? Like astrocytes, glia, immune cells, and so on. So there is this new movement, and I think we need to try to get the right people together. I am hopeful But this is, Vivian, if you think about this like from the business standpoint, I am not sure, but I'm
No.
willing to push this as much as I can to see whether we can see the light. I think is incredibly exciting.
Yeah, I mean, I think the problem, as usual, is that the clinical trials, especially a big one, is what the big expense is, and that's where you need the funding.
Right. And and what you find difficult also, Jason, is parents, right? So the paper come out and you have a bunch of emails from parents and send me the strain, send me this, send me that, right?
Right.
I am very careful, to be honest with you. I know that there is other people who want to go and trying to make money out of this and and sell stuff. I am very careful, right? Because first I'm not sure whether we have the best horse yet, right? Can we create a better strain? So I'm still hopeful that we can create a better strain.
Yeah.
Now, if you think that you can create a better strain, why to run the trial now, right? Because yes, as you said, this is a big expense, right? So
Yeah.
so those are the things. But on the other hand, is you have the necessity of the parents, right? That they are desperate for a potential treatment, and they do anything and everything, right? And I am totally understand it. I fully respect it. I feel for them. I am affected by this as well, in family or very close family. So all I'm saying is, but the more important thing is we need to be responsible, and we need to just to partner with the people who had a vision that we can change, have an impact in society and in human health. So this is the partners that we are trying to find. We haven't found it yet. We have an idea what are the doors that we have to knock, but we haven't found it yet.
So trying to find the gene, is that something you're doing in your lab, or is that not happening now?
Yeah, so this is tough, Jason. Do you remember the two strains I told you? One works, the other one doesn't work. 2,200 genes in total, each of the strains. There's 400 genes which are different.
I see. Yeah.
So 1, 2, 3, 4, 400 genes, right? And unfortunately, I don't have a high throughput screening. The way that we were screening was with behavior. So a pain in the neck. So I have to go. So, okay, let's go back. I have to go. I have to create single gene mutations for all those 400 genes.
Yeah.
Go to church and pray that there's not going to be a combination of genes. Go to church and pray that it's not a SNP, which is just presence of absence.
Yeah.
Right.
Yeah. That and screening mice behavior that would take forever.
Well, the poor student in my lab who did it, and I love that he did it because he took it to heart, and it took forever. And we gained some information, right? Probably he screened close to 20, 30 different strains, right? And everything that we do it at least twice, right? Just to gain confidence. And if you have one that works and the other one that doesn't work, we go to a third time. So a third time you do the behavior with that strain, right? Just to gain confidence, right? Because then you will do your analysis of genes presence and absence. So the idea is that, anyhow. So it is complex, but this is something that I think we are still pursuing. We want to get to the gene, and I think that would be a good chapter. Closing the chapter, I think that would be awesome.
Mauro, instead of screening behavior, which takes forever, have you looked at something in the blood that might correlate with social behavior, like the biopterin that you initially found, or like just oxytocin, something like that?
Yeah. So we are still trying to find better ways. The best model would be something in vitro, right? So then you screen in vitro, right? Because now you have a library, transposon library, whatever, right? So now you have as many bugs as you want, and now you can screen for gain and loss of function, whatever, right? So that would be the best way. We have failed so far. We haven't found a way to screen in vitro. Okay. Through this analysis that I mentioned to you, this very low throughput, we came to the conclusion that there might be a few clusters that we can target, and that's what we are targeting. And we are starting some collaborations with experts at Berkeley, which they have the expertise to do editing in the microbiome in a much efficient way that we do. So we are trying to build into this and leverage that expertise. That would be awesome, and even for all the molecular studies, as you can imagine, Jason. If you just put those two bags and you do say metabolomics or whatever, single cell, whatever. Oh, sorry, what is this?
He's like, that would be amazing, and the phone responds.
What is this?
Like, yay!
This is my Italian's hands, So, so what happened is that if you do genomics or metabolomics, the difference are massive. Even those 400 genes are massive. The difference. Imagine if you just have one single mutation, so your wild type and your mutant, and now you run all those analyses. They could be much cleaner. So I tell my people that sometimes in science, the objective is, in order to find the light, is to reduce the noise, right? And so basically, if we were to find that gene, not only will give us a translational output in the clinic, but also, in terms of biology, I think we will learn so much. We will get to the mechanism that we have an idea. I think we are much better positioned than many of our other colleagues in the field. But by definitely, this is not the entire story. There's no way that this would be the entire story.
Right. Yeah. I mean, I think that's where, you could bypass the this whole issue with the genes if you knew how the circuit itself was being modulated or what activated. Yeah.
Right. Yeah. But this is where you gain little by little. This is where you gain trust. But if in the meantime we can help kids, I think that would be awesome.
Yeah, absolutely. Very cool. All right. Any other questions, guys, for Mauro?
Many, but they're all, did you look at this? Did you look at that? So maybe there's a different forum for those.
No, and the answer is no yet to all those
Not
questions.
yet. Yeah. Well, yeah. I mean, I was going to ask if I was looking at trying to figure out what the exclusion and inclusion criteria were for the trial, which I know is probably not something that you just like have that you necessarily know in its entirety. But one thing I wondered was whether there were people lost to COVID, not lost like passed away, but they had to be removed from the trial because they had COVID. But I also wondered if there was any information about the children's history with allergy that might have been picked up, because for whatever reason, there's a huge overlap or comorbidity of autism and mast cell disorders.
I see.
And so I just thought it might be, and I was reading about the effect of oxytocin on mast cell degranulation, and the mast cells are in the brain, even developmentally in normal, typically developing individuals. So I know that the paper looked at, ruled out any major changes in the adaptive and innate immune systems, but I saw that mast cells were not on there. So I thought it might be interesting to see if that information was, if any indication of allergy, history with allergy was collected when the patients were being brought on to the trial. And then also if there are any hits that come up in any of the work that you guys do that has to do with mast cells, it might be interesting to look at.
Yeah. But so just to be precise, we rule out the adaptive immune system because when we remove mature, we have a mouse model of autism. We have social deficits. We cross it with another mouse in which B and T cells are immature. So you have very little B and T cells, the RAG2 mice. Incredibly painful experiment but actually I think it was very telling. So now you have those animals and you give them the bacteria. and the bacteria is able to reverse the social deficits, indicating that in the absence of B and T cells, the bacteria still can reverse the social deficits. We haven't touched the innate immune system. The reason why is because we don't know enough. Well, I don't know enough about anything, but I'm saying that.
None of us do.
But you
We're
know
all
what I mean? It's
clueless.
like what you do is you do type 1. How do you would you remove one of the interferon receptors, master regulators, IRA3, IRA7? Well, what do you do, right? And each of those experiments need to be done in the background of a knockout for this genetic disorder, right?
Mhm.
Incredibly painful experiments.
Yeah.
Very telling if you have a good answer. But then the other problem that you might have is that if you know that you don't have an effect, is that I always tell my students, right? So if you cut the legs of a mouse, there's nothing that you will do to make it run faster because it has no legs, right? So if you remove exactly. So how can you, right? So
Yeah.
in this case, I think we are still exploring. As I said, we have an idea. We can make a cartoon of how things work, but I think the beauty of biology is that is so we are not like artists, right? That you just have a board and you make your piece. So evolution has done all those experiments for us, and
Yeah.
what we are doing is we are trying to discover it, right? We are trying to undig that information, right? And it's through experimentation and will and a lot of cojones but that you get fascinated by now a bag which is able to change a behavior in kids, which
Yeah, I mean, I
still
think that part
amazing.
to me is fascinating. That obviously these bacteria over time have evolved to modulate behavior in all sorts of ways, and that's what we're trying to discover right now. Is there that, and we didn't know this before the bugs are so integral for brain development, immune system development, let alone these specific behavioral outputs. So it's pretty crazy.
And as I always tell, there's a romantic aspect of this, right? So viruses or bacteria, you know about this, right? So what they do is they hijack the system. They hijack the whole system to be able to perpetuate themselves, right?
Mm.
What a better way for a bug that has to be passed along from one individual to the other, the hijack
Yeah,
social
exactly.
circuitry to
Exactly. That's what I
promote
was
social
thinking.
behavior. So now can be passed along over generations. So this is also super cool, right? If I were to be doing this, is making us social, and there's a lot of implications, right? I mean, if this were to work, I mean, I think um, uh, um, the postdoc who started this work in at Baylor, she came out with a paper in which she gives this when the moms are pregnant in a high fat diet. And when she
Mm.
does this, she's able to reverse the social deficits in the offspring. So, given that this if this were to be safe, can be this generalizable, give it to everyone, right? And see, other social deficits which are not autism driven, right? There's a lot of people when they get old, you know what I mean? There's a massive
Get
social.
cranky.
Yeah, Jason did
Yeah,
a paper on that a while ago.
did you?
About cranky old people?
About
Or just
old people not liking social interaction old mice don't not liking social interaction anymore, and you can open a critical window by giving some LSD or some
Oh, that
psychedelics.
would be fun.
Oh, I thought it was just that
But
they needed to eat more fruit or something. I heard that on the news when I was a
Yeah.
kid and then told my dad he needed to eat more fruit because it would make him less
because it has
cranky.
the L-reuteri in the fruit, perhaps.
Yeah, I mean,
Right,
this
maybe.
idea of evolution. I mean, I'm fascinated because it goes back hundreds of millions of years. I mean, I think these viral genomes that are now all over the genome, the modern genomes, they've propagated themselves also because they've been able to not just increase the survival of a species, but also the behavior. So, it goes back a long time.
Yeah.
Mhm.
So there is a paper from in 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 Scientific Report 2018 from Murillo and Rincon that essentially so Hydra is one of the first animals, right? I mean, it's 500 million years ago, and this animal has a contractility movement, right? It's like a gut essentially, but for the animal, this is the behavior that the animal has, right? And what can you do? It's like 4 cell types, right? And that behavior is driven by neurons. If you remove the microbes in this animal, you affect that behavior and osmoregulation and so on. So 500 million years ago, already microbes were talking to neurons to confer a behavior in this very simplified organism. Now we are discovering that they could be used as a vehicle to change behavior in humans, which I think is still fascinating.
Yeah, I mean, surely microbes is the part of the environment that kind of eukaryotes evolved out of. So it's always been in the environment that we have to kind of navigate and sometimes use. It's just part of us.
Mm-hmm.
Do we know why this particular strain, you said it was difficult to find in the general population? Do we know why that is?
So we don't know.
Or like when it disappeared?
We have speculations of why that happened. So this bug is very sensitive to, say, diet and other conditions. So very interesting. I have an hypothesis that so it has 2 prophages, Jason, and when you have a change in homeostasis of the system, those prophages, for the audience, they will be like equivalent of a retrovirus like HIV, which is dormant.
Okay? Right.
But then when there is a stress, now those prophages pop up.
Yep.
And kill. Oh, again, you see, pop up.
That's so funny.
That was so intentional.
I don't know if that's going to be in the recording, but
this is like, oh my God, this is AI. Anyhow, so they pop up and actually they kill the bug. So they may have a sensor, right? They're exquisitely sensitive to those changes. What other changes or what else can they sense? But, could be diet. We thought also that, stress, right? There's something that, has accumulated over.
Persistent chronic
But you
stress.
put high-fat diet, and this is how we found it, right? You put high-fat diet and other people also, you eliminate it from the population.
Well, that's what I was thinking. I was like, time to talk about like the obesity epidemic and all of that. And just the food industry and how it's changed probably so much about us, not only our guts and our adipose tissue, but potentially also our brains. And what was it like in the last like 30 or 40 years, the frequency of autism spectrum disorder diagnoses has increased. And I'm like, well, that tracks very much from like, when diet started to shift so much, at least in Western culture, towards high fat and high carb diets.
Yeah.
I mean, there's so many correlations, right? We don't know.
True. Correlations.
We don't know whether this is because we have more accurate ways to diagnose it now or so on. Everything correlates, right? The economy,
I was going to say,
yeah.
There are children who benefit from, children who have, I believe there are children on the autism who have an autism spectrum disorder or, related, like, comorbid epilepsy, who benefit tremendously from a high fat diet, but maybe it's the kind of fat. So I think that's a whole nother branch, right? To understand how the certain potentially helpful bags of chemicals interact with diets in a certain context. And it's not a great time to talk about how the NIH, budget for this coming year, it's just not as big as it used to be. So it's may not have the opportunity anytime soon to do that research. But
okay, I'll with that I'll wrap up here.
On that really a positive note.
On a positive note.
It's a call to action.
Readers can or listeners can find the show notes at microbe.tv/TWiN, and you can send your questions, comments to TWiN@microbe.tv. Hopefully, we'll get to some of the questions that you've been sending in. And then consider supporting the science shows of Microbe TV at microbe.tv/contribute. Thanks, Mauro, for joining us. That was it's always fun to have the architect of the work. Thanks, Tim.
Thank you, and thanks a lot, Mauro. It was very enjoyable and good to see quick bench to bedside. It's very impressive.
Thank you guys for coming. Always a pleasure.
And thanks, Vivian, for joining us.
It's fun, as always.
I'm Jason Shepard at the University of Utah. You can find me also on Twitter at Jason Synaptic, or I guess X these days. You've been listening to This Week in Neuroscience. Thanks for joining us, and we'll be back next month. Thanks, everyone.